Bacterial chemotaxis by Parsek. Bacteria have a structure called flagella that help them swim. In E. coli, it has many flagella, and when you spin them counterclockwise, they cause the bacteria to swim forward. If you spin them clockwise, this causes the bacteria to tumble and reorient itself. E. coli is pretty fast. It can move about 18 body lengths per second. As a reference, cheetah at top speed can move 3 body lengths per second. The structure of the flagellum has 2 parts. The basal body has many rings and anchors the flagellum to the membrane. The basal body has motor proteins that cause the basal body to spin. The second part is the flagellar filament, which is made up of subunits of the protein flagellin. I'll be using a fleecy mutant in my experiments. This is a gene that encodes flagellin. The flagella filament is helical and kind of works like a boat propeller. Proton motive force provides power to spin the flagellum. If you look at the top diagram, proton motive force results from a bunch of hydrogen ions that build up on the outside of the cell, purple circles in the diagram. These hydrogen ions want to move inside of the cell but can't because of the membrane. This is like water that wants to move across a dam but can't. By letting hydrogen ions inside of the cell, the bacteria uses the energy to spin the flagellum. This is just like when you open a hole in a dam and harvest the energy of the water flowing for hydroelectric power. On the bottom, you can see that the MOT proteins form the channel that lets the hydrogen inside of the cell. As hydrogen moves across the membrane, this causes the basal body to spin. My research project is about chemotaxis. This is a way bacteria can find their way to food and get away from harmful chemicals. We use our eyes to find food, but bacteria don't have eyes. Imagine if you closed your eyes and tried to find a freshly baked pie in the kitchen. Using your sense of smell, you would find it eventually, but you probably wouldn't walk straight to it. The same is true for bacteria that can sense gradients of food sources. To get to the food source, they use what's called a random biased walk. By alternating smooth swimming and tumbling, they slowly make their way up a gradient towards the food source. Chemotaxis requires a bunch of proteins in the cell. One is called the MCP. This protein sticks outside of the cell and senses the chemicals called either chemoattractants or chemorepellents. The MCP then determines whether the protein KY is phosphorylated or not. Phosphorylated KY causes the flagellum to spin clockwise and cause tumbling. So if the bacteria is swimming towards a chemoattractant, this is going to favor unphosphorylated KY in smooth swimming. I circled the protein KB. I have a mutant strain unable to make this protein. This strain can still swim fine, but it can't chemotax. I will be using this strain in my experiment. This slide is showing how KY interacts with the basal body and influences what direction it is turning. There are different types of gradients bacteria can move to. Some bacteria can move towards oxygen. This is called aerotaxis. Some bacteria can move towards light. This is called phototaxis. The pond water bacteria in the top part of the figure can move towards different wavelengths of light, produced by a prism. The bacteria on the agar plate below, called Rhodococcus, can move across the surface of the plate towards a light bulb that it's sitting next to. I'll be using 3 strains of bacteria: a wild-type strain of Pseudomonas aeruginosa, a fleecy mutant strain of this that's unable to make a flagellum, and a key B mutant strain that can't chemotax. My main hypothesis is that the 2 mutants will be unable to either swim or respond to chemotactic gradients. My first experimental system involves swim agar. This agar is poured into plates and bacteria are inoculated into the agar with a needle. The bacteria grow and can swim through the agar. When bacteria first start to grow at the inoculation point, they consume nutrients, creating a gradient of nutrients, with higher levels at the areas outside of where the bacteria grow. This should cause bacteria to chemotax towards the exterior of the inoculation zone. A chemotaxis mutant should be able to swim but unable to chemotax, so it should spread slower than the wild type. The fleecy mutant is stuck because it can't swim. That's exactly what I saw on the two nutrient sources, glucose and LB, which is just many amino acids. The second experimental system involves a plug plate assay. Here I take bacteria that are present in swim agar and pour a plate with the bacteria already present in the molten agar. The agar solidifies, and then I add agar plugs into the swim agar that contain high levels of potential chemoattractants. The bacteria can swim through the agar towards the plugs. I saw for the wild type that the bacteria swam and formed a ring around the glutamate plug. A slight ring formed around the glucose plug. The chemotaxis mutant formed a faint ring around the glutamate plug, which was a bit of a surprise. The last experiment didn't work well. It involved a confocal microscope and fluorescent Pseudomonas. In this assay, you place a chemoattractant into a capillary tube that you then stick into a Pseudomonas culture using the microscope. You can visualize bacteria swarming around the end of the capillary tube. We have many problems with this experiment. The device leaked, and the bacteria did not fluoresce. In summary, my results showed evidence for bacterial chemotaxis in the first two experimental systems. The chemotaxis mutant was poor at responding to chemoattractants in these assays. As expected, the fleecy mutant was a great negative control. These are the resources I used in my presentation. Thank you for listening.